Welcome to Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health, where we bring evidence, experience, and perspective to make sense of today's leading health challenges. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes.
This is Grace Asiri. In honor of World Mosquito Day, which is observed annually on August 20th, we bring you an extended episode of Malaria Minute, a podcast from the Johns Hopkins Malaria Research Institute. Let's listen.
Over 125 years ago, scientists made a landmark discovery that female Anopheles mosquitoes were the vectors of malaria, that they carried infective parasites from human to human, inflicting illness and even death upon most they bite. To stop the consequences, scientists figured you first need to stop the mosquito. Over a century later, female Anopheles mosquitoes remain the target of malaria control and prevention. Bed nets that you sleep under, or sprays that kill the mosquitoes around the home, have become trusty tools. Now, a new tool called gene drives offers a different approach. They involve genetically modifying the mosquito so that they can't transmit the disease. This special episode of Public Health on Call explains how they work and how they might fit within the broader context of malaria public health efforts. We ask, what are the challenges of moving gene drives from the lab to the field? How can researchers ensure that gene drives are safe? And how can the concerns of local communities be addressed? The story is told by experts from different disciplines from different countries, all working together to make gene drives a reality. We start with Professor Anthony James of the University of California, Irvine, who describes the novel idea of genetically modifying mosquitoes.
Well, the idea is actually very old. What we didn't have was a system that will allow us to make it work. But with the discovery of the biology based on the CRISPR system, people recognize that this is something that could work very quickly.
The CRISPR-Cas9 system was a turning point for the genetic control of mosquitoes. This gene-editing technology lets scientists cut and paste genes into DNA with speed and precision. It's the key that enables gene drives to work, and to understand this, we need to go back to basics.
Even though it is very simple genetics, it is complex, and not everybody's taken a genetics class. What I tell people who don't really understand the biology is they know that their bodies are made up of cells and that those cells have chromosomes in them, the chromosomes that contain a lot of information that's useful. Now, when cells are dividing, it's really important to them that their chromosomes are intact. If you break a chromosome, the cell is going to stop everything it's doing till it fixes that chromosome. And we take advantage of that.
Take, for example, cancer treatments. Cancer cells grow without limits. If we can cut the chromosomes with radiation or chemicals, we can stop the cancer cells from growing. The simple idea is that a cell will stop everything until it fixes its chromosomes, and the CRISPR-Cas9 system allows scientists to cut a chromosome in a very specific place, and then the chromosome looks to repair itself.
What we do is we trick the cell. We cut a chromosome, and then we inject millions of little pieces of DNA that look a little bit like that chromosome, so that when the chromosome goes to fix the cell, it uses what we put in there. And now it brings in something that we want.
You can remove genes that the parasite needs to develop and to live.
The parasite's no longer going to be able to grow.
Or you could add a new gene entirely.
Construct a gene that has all the properties that you want. So it's a novel gene. And this gene is designed to impact parasite development, prevent it from being transmitted.
All of those modify the mosquito, but you can also introduce or remove genes responsible for fertility and make the mosquito sterile, known as population suppression. That's how you edit one mosquito, but the real power of gene drives is inheritance. Because CRISPR-Cas9 is a bacterial system, it spreads to the tissues that make up sperm and eggs, called the germline. In a fertilized egg, the CRISPR-Cas9 system edits the DNA inherited from the partner that doesn't have the gene drive. This means that over multiple generations of mosquitoes, more and more of them will have the modification. And this is called biased inheritance. So that's the theory, that by interfering with chromosomes you can modify the DNA and make the modification spread. But why would you want to do this in the first place? What's wrong with the existing tools? George Demopoulos is a Deputy Director of the Johns Hopkins Malaria Research Institute at the Bloomberg School of Public Health.
All the currently used control methods for malaria requires a continuous effort and compliance by the endemic population. So it becomes very costly and labor-intensive to perform malaria control today. And what we really need is a technology that wouldn't require that involvement, that continuous involvement and investment in malaria control. And that's why genetic modification of mosquitoes has become an attractive alternative approach for malaria control, because you would basically make mosquitoes resistant to the malaria parasite and stop the transmission in the mosquito.
Part of the work of his lab is to identify useful genes that could be used in a gene drive. His lab looks at what happens in the mosquito when it's infected with malaria, with the aim of improving its immune system and building a better mosquito.
The mosquito has its own immune system, which is using to fight the malaria parasite. And some of the immune factors or genes that are part of the mosquito's immune system are actually quite effective at blocking the malaria parasite. So one approach to develop these malaria parasite-resistant transgenic mosquitoes is to identify such genes.
Another approach is to introduce entirely new genes that the mosquito doesn't encode to try and stop the parasite progressing in the liver. One recent example, tested in the lab, called AGTP13, has multiple functions. It binds to stop the parasite progressing in the mosquito and also kills the infective form. As George makes clear, developing a gene drive from identifying a target gene to releasing them into the wild is a huge undertaking and requires multiple specialized labs.
A gene drive-based population modification approach, where we modify a malaria-transmitting mosquito population to one that cannot transmit the malaria, involves several laboratories. It will involve laboratories that are expert on building the gene drive technology that will propel these resistance genes into mosquito populations. Other laboratories will develop the actual locking mechanisms in the mosquitoes, and yet other laboratories will perform testing. Other laboratories will specialize in modeling. Other groups will address the social and political aspects of this technology, which is very important to engage the endemic population and stakeholders in this technology and make sure that they understand it and they are on board and they're actually participating in implementing and deploying the technology in their countries.
George is optimistic. He doesn't think that gene drives will, on their own, be appropriate, but when applied in an integrated way with other tools, he thinks they could be a powerful tool for reducing cases and deaths.
The most important step now is to engage the endemic countries in this technology. There is some engagement and training steps that will have to happen before we can start the real releases.
We now turn to Kenya to speak with Damaris Matoke-Muhia, a principal research scientist at the Kenya Medical Research Institute.
Kenya, as many other African countries, is part and parcel of the discussions going on to identify novel approaches to fight against mosquito-borne diseases. The communities are asking, what are the scientists saying? What is the science telling us? What are those findings? Because they are very keen to have a technology that is user-friendly, that is not going to bring harm. And they believe that if the scientists have the data and they are well informed the data, then it's a tool that they can adopt.
As with any new technology, there are some concerns and fears.
There is a question that what if this mosquito comes and becomes a super mosquito that's maybe now starts transmitting other diseases, maybe not transmitting malaria, but now it can be able to transmit other diseases which are more harmful. What if this mosquito affects our ecosystem? What if actually it does not reduce malaria? We know some of the answers, but we don't know others. And that's why there's continuous research. And that's why continuous engagement with the communities to hear and to know their perceptions, so that even their fears, so that we can also say from data, this is what we face. We don't have all the answers yet as even research scientists. And that's why continuous research and even wanting our own local research. But more so, we have the confidence that some of the claims by the communities are likely not to be, we are likely have the answers to them, but there is need for continuous support on research to mitigate against them.
Damaris says there's goodwill from the Kenyan government and scientific research community to bring gene drives to fruition. She says they just need everyone on board. What's more, there's a regulatory framework in place to follow for ethical approval. The National Biosafety Authority in Kenya, which has been used to make decisions around GM crops like cotton, can also be used for gene drives. But unlike GM crops, which can't spread because they've been made sterile, gene drives designed to spread as far as mosquitoes fly do, posing challenges for regulatory approval. There's agreement that gene drive research should be done in a phased fashion to make certain the technology is safe and effective in the field. It'll be some time before gene drives are released into the wild, but the public health payoff could be great. A modern twist on an old public health objective to stop the cycle of malaria transmission in the mosquito so that humans can't get the disease.
Public Health on Call is a podcast from the Johns Hopkins Bloomberg School of Public Health, produced by Joshua Sharfstein, Lindsay Smith-Rogers, Stephanie Desmond, and Grace Fernandez-Cesiri. Audio production by J.B. Arbogast, Holly Cardinale, Spencer Greer, Matthew Martin, and Philip Porter, with support from Chip Hickey. Distribution by Nick Moran. Production management by Catherine Ricardo. Social media run by Grace Fernandez-Cesiri. Analytics by Eliza Rosen. If you have questions or ideas for us, please send us an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Thank you for listening.